me?
I can, yeah. Thanks, Sam.
All right.
Okay, so thank you again to everybody for attending today. I really appreciate that you're here to listen to my presentation. And also thank you, Sam, for that wonderful introduction. So the 4 W's of microcirculation and tissue pO2. So today we're going to be discussing where these measurements are possible for in vivo animal model experiments, when these measurements are appropriate, why measuring both tissue pO2 and microcirculation or microvascular perfusion are both so important, and what to use to measure them. So closer to the end of the presentation, I'll be showing a couple data examples so that you can get a feel for how others have used these measurements in the past with the particular systems that I'm going to chat with you about today, and then also to give you some ideas for how they might work for your own experiments in the future. So when we're talking about tissue pO2 and microcirculation, we're really talking about the capillary beds in the tissue, where the functional tissues and organs are receiving oxygen and nutrients from slow-moving red blood cells, and measuring the perfusion and oxygenation in these tissues and how it's a measure of tissue vitality. So why is it important to measure both perfusion and tissue pO2? Well, it's widely known that the survival of tissues and organs relies on an adequate supply of oxygen and nutrients. The measurement of tissue oxygen tension, or PTiO2, provides a direct measurement of the balance between oxygen supply by the blood and metabolic oxygen consumption by the tissue. In other words, a readout of oxygen availability at the cellular level. Research continues to show that heterogeneous changes in perfusion of organs leads to areas of hypoxia and organ dysfunction. Monitoring these changes is essential in order to determine the pathological mechanisms leading to tissue hypoxia and the subsequent development of potential therapies. When doing in vivo experiments, understanding the conditions that the cells are experiencing in the tissue is going to be very important for understanding how the tissue will respond to certain treatments and interventions. Using tissue vitality, you can also monitor and quantify tissue injury and recovery, if there is any, from therapies and treatments. So when should you be monitoring tissue vitality? This is highly application dependent. However, in most cases, measuring the oxygen and perfusion should happen before, during, and after any treatment or occlusion. So when we get to data examples later, you'll see that having these measures and watching them change in real time is a fantastic way to understand what conditions the cells are experiencing in the tissue of interest during whatever treatment you happen to be doing. So examples of these experimental designs can include pre, during, and post ischemia events, including, but obviously not limited to, MI surgeries, stroke models, aortic and arterial clamps, things like that. The oxygen monitor that I'm going to discuss with you later can also be used on its own for in vitro type work to measure oxygen concentrations in perfusate buffers for ex vivo Langendorf heart preps, for example, and also in combination with the flow monitor that we'll discuss later as well for ventricular wall muscle tissue vitality in a Langendorf prep as well. So they can be used in combination there, which is really cool. And then another great application is tumor hypoxia in cancer research. So measuring the oxygenation or the hypoxic regions and the perfusions of these different regions for heterogeneous tumors. So to understand exactly where blood is delivering oxygen and nutrients and where it's not, and maybe potential sites in the tumor that are going to be maybe better for treatment delivery. So these are just some examples of kind of when you can measure before, during, and after treatments. When I think about the when component of this sort of webinar, I'm also thinking about controls. So an important aspect of measuring tissue oxygen specifically is ensuring that the experiment has these controls. So when measuring real time with an animal under anesthesia, or perhaps using perfused organs, by varying the concentration of oxygen in the air that the animal is breathing or in the buffer that is being used to perfuse the organ, you can do what we call an oxygen challenge. So this means that you should always have the animals breathing normal levels of oxygen, either medical air or 21% oxygen balanced gas is recommended. But to ensure sensors are functioning properly, you can increase the oxygen concentration and watch the tissue oxygen increase on the trace. And you'll see a couple of these traces later in the presentation. But then once you see that the sensors are responding, you can turn that oxygen back down to normal or 21% and start your experiment. And this is going to ensure that all changes in the tissue are physiological and not a result of poor experimental conditions. Something that I've come across in so many experimental designs and demonstrations of these products is that the animals are actually breathing 90 to 100% oxygen during procedures where researchers are trying to measure in vivo pO2 changes. So the values are just way too high when they're breathing 100% oxygen to really be considered in vivo and realistic. So definitely something to take into account when you're planning your studies, especially regarding tissue oxygen. So that kind of takes care of the when should you be measuring, but where can you measure tissue vitality? This really depends on what tissue you're interested in and what your application is. However, these measures can be made in almost any tissue you can think of. So some of these examples on the screen are more common applications. However, we do have some more rare applications in our citation list that you can find on our website after the webinar is over. So you can take a look at that extensive citation list for these systems and get an idea for where others have been using these systems in their research. So it's great that you can measure in many tissues, but when I think of the where component of this webinar, again, I'm brought back to the importance of having controls. So it's important to have a sensor in the area that you're interested in, sure, but it's also important to have a sensor in an area that maybe you're not so interested in measuring as a control. So what this means is that if you're interested in kidney pO2, it might be a great idea to also have a sensor in skeletal muscle or skin perhaps to monitor changes in a different organ over time. So if you do an oxygen challenge with an animal under anesthesia and bring the oxygen up to 100% before you begin your experiment, if you have multiple sensors in different tissues and organs, you can monitor the time it takes for those different tissues to show the pO2 increases. And you can also use it as a confirmation that the tissue pO2 and perfusion changes in the tissue of interest are physiological and not just a global change in oxygen saturation or decreased heart rate that's affecting every tissue regardless of your treatment. So an example of this is, like I said, if you do have a sensor in the kidney and then also one in the skin, and you turn the oxygen up to 100% that the animal is breathing, that's great, and you'll see both sensors respond, but you'll probably notice that the one in the skin is going to respond a little bit slower because it's less of a quote unquote important organ and it's not perfused as highly as the kidney is. So these are some things that you can kind of think about when you're planning your studies. Another example is in stroke models. So sure, the affected hemisphere of the brain is really important to you as a stroke researcher. However, it's a really great idea to have sensors in the unaffected hemisphere as well. That way you can get a full picture of what's going on in the brain, just to ensure that whatever treatment you're applying isn't affecting both hemispheres, or that it is affecting both hemispheres, and maybe that's something that you want to kind of tease out of your experimental design. So we've briefly covered the when and the where and the why you should be measuring perfusion and tissue oxygenation. But let's dive into the what. So what can I, as a researcher, use to measure tissue vitality? So the tissue vitality monitors I'm going to discuss with you today are the OxyLite and the OxyFlo systems by Oxford Optronics. They are in the bottom right corner of your screen, and these systems stack together to give you a fully plug-and-play tissue vitality monitor that uses laser Doppler perfusion measures combined with state-of-the-art oxygen sensing technology to give you multi-channel and easy-to-use tissue vitality monitor. So to discuss all the amazing features of these systems, I'm going to talk about each one individually, and then I'll bring them back together to discuss why these systems are so versatile. So let's start with the OxyFlo. So the OxyFlo is a blood flow monitor, And as I mentioned, it uses laser Doppler principles, and it works best in acute models where blood flow is being altered during the experiment. So in occlusion and vessel constriction models or clamping models, this system will provide a great way to monitor the changing tissue perfusion. There are 3 different configurations for both the OxyLite and the OxyFlo that you can kind of choose from. There's single channel versions and then 2 and 4 channel systems, meaning that you can use up to 4 sensors at any given time if you choose a 4 channel system. So the probes for the OxyFlo can be invasive or non-invasive depending on your tissue of interest. And Oxford Optronics has developed a proprietary technology to reduce some of the cyclic movement artifact that laser Doppler users are so familiar with when doing in vivo animal studies. So things like respiration rate and the moving of the diaphragm and the rib cage does cause cyclic movement artifact. And anyone that's used a laser Doppler has probably experienced this. And so Oxford has developed this proprietary artifact rejection technology to help kind of filter out some of that noise, which makes the system a lot easier to use, and you don't have to necessarily account for that later when you're doing your data analysis. So how does the OxyFlo really work? So the OxyFlo utilizes the well-established method of laser Doppler flowmetry, or LDF, to provide continuous measure of tissue blood flow. So LDF works by illuminating a small region of tissue under observation with a low power laser light from a sensor containing optical fiber light guides. So the 785 nanometer laser light for the OxyFlo from one fiber is directed towards the tissue, and it's scattered within the tissue, and some of the scattered laser light is incident upon the moving red blood cells within the tissue, and this light becomes Doppler frequency shifted as a result. A second optical fiber, here labeled the receiver, collects that backscattered light, and the Doppler shifted light from the tissue also returns to the photodetector within the OxyFlo actual box. And this photodetected signal comprises a broad spectrum of Doppler frequency shifted signals, which are generated as a result of the movement of the red blood cells within the tissue. The microvasculature and the blood flow in there is electronically calculated as the product of the mean red blood cell velocity and mean red blood cell concentration in the volume of the tissue that's under illumination from the sensor. So in shorter, more brief, succinct language, essentially the light is bouncing off of the red blood cells back into the system. It's being photodetected and averaged together from the whole sampling region to give you an arbitrary measure of blood flow. So that's how the OxyFlo works, and we're going to move on briefly to the OxyLite. And so OxyLite monitors are intended to measure partial pressure of oxygen, or pO2, in aqueous conditions. So these are conditions of 100% relative humidity, or basically liquid or tissue. So the oxygen partial pressure is often referred to as dissolved oxygen and is very much distinct from the measurement of hemoglobin oxygen saturation using spectroscopic techniques like pulse oximetry. And I'll show you a little bit more about that and how they're compared to each other later. So the OxyLite monitors also optionally provide a real-time readout of temperature from the sensor tips. So that can be a great way to monitor the temperature of your tissue, but then also to compensate for different temperatures in the oxygen reading. And I'll get into that in the next couple slides as well. So the OxyLite, how does it work? So the fluorescence-based technology employed by the OxyLite provides an absolute measurement of dissolved oxygen in either millimeters of mercury or kilopascals. And you can set that in the settings. In tissue monitoring applications, this provides a direct readout of the balance between oxygen supply by the blood and oxygen consumption by the tissue, or a readout of oxygen availability to the cells. So this contrasts the blood oxygen saturation assessment, or pulse oximetry, that I mentioned before, which merely describes the hemoglobin oxygen status. And so here's the comparison chart between the OxyLite and how it works and the pulse oximetry technology. So they are definitely different technologies, and they both have their time and place, but one of the main kind of differences is that the pulse oximetry is a whole body measure. So this is measuring how oxygenated the blood is in any region of the body versus the OxyLite, which measures a single area of interest. And so it's tissue specific, and it's not really a whole body reading, and it's really more of the site of interest. And so how exactly do these OxyLite sensors work? So like I said, the OxyLite is an optical device, and the sensors are based, or the science behind these sensors is based on the principle that the presence of molecular dissolved oxygen in tissue or liquids can terminate or quench light emitted by a fluorescent compound. So in this case, the fluorescent compound is the dye in the sensor. So the quenching of the fluorescent light is proportional to the pO2 in the vicinity of the dye. So with the OxyLite sensors that are plugged into the OxyLite box, short pulses of LED laser light are transmitted along the fiber optic sensor to excite a platinum-based fluorophore dye bonded to the sensor tip. And so the resulting emission of fluorescent light, quenched by the presence of oxygen, is detected by the instrument. So the instrument measures the lifetime of the fluorescence, which is inversely proportional to the concentration of the dissolved oxygen, and it's interpreted to provide an absolute value for oxygen in millimeters of mercury or kilopascals. And the calibration information for each sensor is actually stored in the sensor connector, which is proprietary to Oxford Optronics. And this allows them to be fully plug and play. So they are pre-calibrated back at the facility. They go through rigorous testing, and this calibration information is stored in the sensor so that when you plug it into your OxyLite, you don't have to worry about doing any sort of zeroing or any sort of calibration. You just plug them in and you can start your experiment. And so with that, the OxyLite monitors also perform automatic temperature compensation. And so they can generate a continuous readout of temperature when combined oxygen temperature sensors are used. And for a full list of sensors that are available from Oxford Optronics and from Scintica, you can visit our website after the webinar and sort of head to the sensors tab, and you'll see a huge selection of sensors that really make the system so versatile, and you can use them for whatever application you have. So now that we've kind of covered both the OxyLite and the OxyFlo and the technology behind them, we can talk about how to combine them for the ultimate tissue monitor. So when systems are stacked together like this, our combined sensors can be used. And so these combined sensors have a red connector and a blue connector that plug into the equivalent OxyLite for blue and OxyFlo for red. And these sensors allow you to measure tissue pO2 and perfusion from one sensor. And these combo sensors are usually approximately 450 microns in diameter, so they allow for a minimally invasive way to measure tissue vitality from the region of interest. We also have something called a triple parameter sensor, so that incorporates perfusion along with the pO2, like I mentioned before, but it also has the temperature sensing technology with a little thermocouple that's wrapped around the bare fiber. And this is our most popular sensor. And many of our in vivo users prefer this sensor as they can get more information with the same number of sensor insertions than just using the OxyLite or the OxyFlo on their own. So pretty soon we're going to take a look at some of the applications for this system and some data examples that were graciously shared with us by some end users and some of their unpublished experimental data. But before we do that, Sam, I will pass it to you for a quick audience poll.
Thank you, Sarah. I will let you take a quick break while we launch the next poll. What is your organ system tissue of interest? I know we have quite a variety of attendees, and we kind of would like to know where is your area of interest? Is it the brain, cardiac, kidney, liver, GI tract, ophthalmology, cancer, tumor, skeletal muscle or other. I know that there is a lot of research being done out there, and we just like to get to know what you guys are doing a little bit better. I'll just give you guys 10 more seconds to be able to answer, and it is multiple choice, so if you do have multiple areas of interest. Okay, seems like mostly everyone has answered, and the majority are doing brain research. Okay, thank you. And back to you, Sarah, for the rest of your presentation.
Thank you, Sam. Okay, so it's great that most of you are doing brain because this data example that I'm about to show you is in the brain. So this particular data slide was gracefully shared with us by a group in Germany. And so I will give you a little bit of background on each experimental procedure and then kind of an overview of what you're seeing on the screen. So it can be kind of confusing at first. So this particular experiment was done in an anesthetized rat. And the site of interest here is the hippocampus. And this researcher is looking at pO2, temperature, and perfusion. So after drilling a burr hole, a combined fiber optic probe, which is the triple parameter sensor that I mentioned before, is inserted into a small incision in the dura into the hippocampus. And so the probe was attached to a micromanipulator and positioned at a depth of 4.5 millimeters from the brain, from the skull. And so global ischemia here was induced by withdrawing blood to a target mean arterial pressure of 35 millimeters of mercury over a period of 8 minutes. So that's event 18 at the bottom there with the orange box around it. And then the 8-minute span was to event 39. So this resulted in an almost parallel decrease of microvascular blood perfusion and pO2 to levels which were maintained across the ischemic event. And so ischemia was maintained for 30 minutes, and after which the total blood was resupplied over a 15-minute period, which was event number 26 and 27 in the bottom there. And the hippocampal perfusion and pO2 recovered to starting levels where perfusion recovered slightly ahead of tissue pO2. So that's sort of to be expected. If blood is delivering oxygen, of course, the blood has to come back first before the oxygen will increase. So this is a nice example of how somebody has used the triple parameter sensor in a global brain ischemia type experiment. So I'm hoping that this type of example is giving you guys some ideas of how you would like to measure in your application and your animal model. So again, if you have any questions about any of the data I'm going to show you, please submit them to the Q&A box, as Sam mentioned. So this is another ischemia reperfusion type study and also in a rat model, but this is actually in muscle. And so again, a triple parameter OxyLite OxyFlo sensor was used here, and it's inserted into the leg muscle in a rat to monitor pO2 perfusion and temperature. So in this particular application, an infrarenal aortic clamp was performed. So that's event number 6 here at the bottom, labeled ischemia. And this causes a rapid drop in the microvasculature blood flow and the pO2. And as expected, the blood flow decreased ahead of the tissue oxygenation as the tissue was consuming the oxygen that was already available. And then approximately 7 minutes later, event number 7 at the bottom there, labeled reperfusion, the clamp was released and an immediate response in the form of a blood flow overshoot or hyperemia is shown here, the red line, and a temperature increase was also observed. The tissue pO2 level also overshot with the blood overshooting as well, and then it settles back to pre-intervention levels. And so this is sort of a great way to monitor exactly what's happening in the tissue, and also a great way to monitor if the ischemia reperfusion actually caused any injury. And so in a later slide, you'll actually see how you can kind of quantify that injury. So we're going to go back to the brain, and this isn't necessarily an ischemic event, but it is definitely a way to monitor what's happening in the brain in regards to blood flow and oxygenation in response to chemicals. So in this particular application, the OxyLite OxyFlo system was used to monitor the response of cortical perfusion in an anesthetized rat to a challenge of norepinephrine. So following a craniotomy, which was a 6 by 6 millimeter hole in the rat skull, a combined fiber optic probe, a dual parameter sensor, which measures the oxygen tension and the blood flow, was inserted through a small incision in the dura into the cortex of the rat brain. And this probe was attached again to a micromanipulator and positioned at a depth of 2.5 millimeters. So along the top in purple, this is sort of a progression of the changing in the mean arterial blood pressure. And so at a mean arterial blood pressure of 90 millimeters of mercury here, norepinephrine was infused intravenously at event number 23, increasing the mean arterial blood pressure to 130 millimeters of mercury. This in turn results in a sustained elevation of cortical perfusion and increasing in parallel but slightly ahead of the cortical oxygen tension, so just as expected. Following reduction of the norepinephrine infusion to a lower dosage, the mean arterial blood pressure decreases to 115 millimeters of mercury, and cortical perfusion is slightly reduced as well. The cortical oxygen tension actually remained unchanged. And then at this point, they stopped the norepinephrine infusion completely at event number 25 here, and the mean arterial blood pressure drops from 115 millimeters of mercury all the way down to 95 millimeters of mercury over this kind of 4-minute period. And then the cortical perfusion and the cortical oxygen tension decrease steadily, sort of over time, to pre-treatment levels. And so that takes about 4 minutes, and then they start to approach baseline levels, and they'll probably level off later in the data stream. So that's just an example of how you can monitor, not just with ischemic conditions where you're actually clamping or ligating, but really a change in the way that the entire body is responding, and then monitoring that in your specific tissue of interest. So there's definitely a lot of other things going on in this rat, not in the brain, and we don't have any sensors there to monitor that, but this is what's happening during a catecholamine infusion in the brain of this rat. So a really cool way to monitor the changes in perfusion in the brain with catecholamines. So we'd like to thank the group in Germany for that as well. The next data slide that I'm going to show you today is kind of more on our wound healing and plastic surgery kind of side of applications, so a little bit further away from the brain and the muscle applications. So for those of you who are unfamiliar with a pedicle flap, essentially they're used clinically for a lot of the time like burn injuries where the patient is not stable enough to undergo full reconstruction, but that the tissue needs to be protected. And so they'll actually sew the organ that's damaged, like for example, if it's a hand, into an area of the body that's comfortable for the patient, like the abdominal region. And they'll actually sew the limb into and under the skin so that the patient has some time to recover and to heal before they undergo reconstructive surgery. So a really interesting and sort of a niche application that I wanted to highlight. So in this particular application, the OxyLite OxyFlo system is used to differentiate between arterial occlusion and venous congestion in a pedicled groin of an anesthetized rat. So a combined triple parameter probe measuring oxygen, pO2, and temperature is used here to monitor blood flow, tissue oxygen, and temperature in that particular region. And so it's inserted centrally into the pedicled flap. And in the figure, the supplying artery is clamped for 15 minutes. So down here, event number 5. and released at event number 6. And so you can kind of see the drop in blood flow happens immediately. There's a slight drop in temperature, and then following that, of course, is the drop in pO2. And so at event number 6, the clamp is released and the blood perfusion kind of comes back up again, and then slowly also the pO2 here. And then so when we get to event number 7, which is at the top here, just in case you didn't notice it, this is the vein being clamped. And so the vein connecting the flap of tissue to the trunk of the rat is clamped for 25 minutes, and it's released at event number 9 down here. And so the results here show an instantaneous drop in blood perfusion, followed by a slower decrease in pO2 than we saw in the arterial clamping. So that makes sense. And then after the release, the tissue injury can be quantified here because the perfusion does not return to pre-treatment levels. And also the tissue pO2 doesn't return to pO2 or to original pO2 levels. And so that's a really great way to be able to quantify the tissue injury here. And you can use that to sort of stratify your animals if you're doing an ischemia reperfusion type study. So really, really cool. And we like to thank the group in Sweden for this unpublished experimental data. So if you have any questions about this, please submit them and I will try to get to them at the end of this presentation. So our next data slide is a larger animal. So most of the ones that we've gone through today are rats, but I wanted to show you one for larger animals. So this one is done in an anesthetized pig. So the OxyLite OxyFlo system was used here to measure the response of the gut mucosa to changes in ventilation in an experimental pig model. So a combined triple parameter sensor measuring oxygen, flow, and temperature is inserted into the small intestinal wall of an anesthetized and intubated pig that weighs approximately 45 kilograms, just to give you sort of a size reference. And during the experiment, the ventilation is stopped at event number 17 for approximately 1 minute, and then it's reestablished here at event number 18. So the figure below, or the figure shown here clearly shows an immediate drop in microvascular blood perfusion upon cessation of ventilation, followed by a steady increase in blood flow as a compensating reaction to the continuous decrease in pO2. So upon reventilation, the oxygen tension in the gut mucosa can be seen to increase over about a 2-minute period until reaching its original pre-intervention levels. And the microvascular blood flow in the mucosa also reverts to its pre-intervention values. So it doesn't look like there's any sort of long-term tissue damage there based on these results. And this experiment shows the effectiveness of the OxyLite OxyFlo systems in continuous monitoring and real-time changes. So in this particular application, they're doing a global change by halting ventilation on an intubated pig and then watching a very particular organ of interest to them as it changes with the slowing of ventilation. So a very interesting study, and we thank the group in Germany for this permission to show you this data. And my last data slide today is not necessarily an in vivo model, but I did want to highlight that these systems are also really great for ex vivo models and perfused organs that I kind of mentioned at the beginning of the presentation. So in this particular application, since there is not technically blood flowing per se from a beating heart, this is an OxyLite or oxygen only study. But I did want to highlight that the OxyLite system here was used to monitor the tissue oxygen tension in response to perfusing the isolated rat heart with reconstituted blood. So the data on the graph shows the effects of the increased blood flow at event number 1. And you can see that that tissue pO2 increases nicely there. And then also the tissue response to the stopped circulation and how the tissue pO2 drops pretty quickly. And then upon reperfusion, it goes back up. And so a single bare fiber probe measuring oxygen in the right ventricle was done. They actually placed the fiber sensor through the pulmonary artery. And the temperature was monitored by different means, not using the OxyLite, but a constant value was used to calculate the pO2 over the course of the experiment. And the pO2 level in the myocardium responds rapidly to the changes during the experiment. And so this is actually measuring the pO2 in the quote unquote blood in the right ventricle as the flow is started, increased, and stopped. And so the tissue is consuming oxygen from the reconstituted blood in the ventricle over time. And you can see that as soon as the perfusion is stopped, you get a massive decrease in the oxygen. So that fluid is being completely deprived of oxygen, and all the oxygen is being pulled out until the reperfusion is started again, and then the oxygen can kind of come back. So really cool study, and we want to thank the group in the Netherlands for this data here. So a great way to measure ex vivo as well as in vivo the OxyLite system. So that's the last data slide I have to show you today, and I'll hand it back to Sam for a quick poll.
Thank you, Sarah, and we'll give you a second to catch your breath. The last poll that we have for today is what systems from today's presentation do you have the most interest for your research? Please select all that apply. So if you were interested in learning more about the OxyLite, which is the tissue oxygen monitor, or the OxyFlo, which is the laser Doppler flow monitor, if you're interested in both, also you may be interested in a virtual demo or on-site demo where it's possible to do those at the moment, due to the current circumstances. Some on-site demos may not be possible. Or you may just be here for scientific curiosity and don't want to receive any more information for this. So please select all that apply. We'll give you guys about 15 more seconds, as it seems like people are undecided yet. 5 more seconds, and sounds good. Awesome. I'll send this back to Sarah.
Awesome. Thank you, Sam. And thank you, everyone who has attended my presentation. Thanks for taking time out of your busy schedules. I know that you're all have a lot of things to get back to, so I won't keep you any longer with my presentation, but I just wanted to finish with this last slide, kind of showing you the systems again. And if you do have any questions for me about the systems or about any of the data that I showed you today, please, again, submit them to the Q&A, and I will try to get to them, well, actually right now. So we're gonna go ahead and begin our Q&A. So Sam can just give a little explanation of how that works.
Yes, thank you, Sarah. So just to remind everyone, we encourage people to ask questions in the Q&A dialog box, and we will get to as many as we can in the allotted time. So we have about 18 minutes here. So a couple of questions have come in as you were presenting, Sarah, and the first one is, can the laser Doppler system be used in the heart for myocardial perfusion?
Okay, I was expecting a question about this. So the quick answer to that is no. Because the heart in vivo is beating, the movement artifact in that particular tissue is going to be way too high and way too difficult to filter out, especially in small animals like mice or rats who have heart rates anywhere from 350 to 600 beats per minute if they're conscious. And so while the OxyLite is a great tool for measuring tissue oxygenation in heart muscle, the OxyFlo is just can't really be used there. So that's part of the reason why when I showed the cardiac example, the perfused rat heart, it was really just the OxyLite, because the laser Doppler system, just the artifact can't be filtered out at that point.
Mm-hmm. So you mentioned different types of sensors in your presentation. Robert asks, how big are the fiber optic sensors?
So there's a range of different sizes for any of the sensors. I'm not going to go through all of the sensors because there's way too many, but we do have videos on our website under the sensors tab on the OxyLite, OxyFlo, and the combined system pages that give you more specifications of how big each sensor is and kind of what the application it was kind of created for. And you can get all of that information there if you are curious about them.
Great. Another user actually asked about sensors. Can you use the sensors non-invasively?
So for the OxyLite, no. So the sensor has to be within 2 millimeters from any environmental oxygen contamination or air in most cases. So it does have to be 2 millimeters below the skin surface or the organ surface, whatever surface of the tissue is exposed to air. However, the OxyFlo sensors can be used non-invasively if you want. So for applications involving skin or the surface of different organs, OxyFlo does have surface sensors for a more non-invasive approach, which is really cool. So there's a little bit of flexibility there in the non-invasive approach for the OxyFlo, but the OxyLite is always invasive. So thanks for that question. We have another question here, if you don't mind, if I just can kind of read it out loud, Sam.
Feel free.
The OxyFlo, can it be used to measure blood flow in mouse skin? Definitely. So if you wanted to do that invasively or non-invasively, there are options for that. And it works like any other laser Doppler would. But what's really great about it is that you can combine it with the OxyLite if you're interested in measuring changes in flow, but then also combining that with the results of the changes in flow. So whether oxygen is increasing or decreasing with the increased or decreased perfusion. So yes, you can definitely use the OxyFlo to measure in skin. Another question here is, oh, sorry. They're scrolling away from me. Is there any possibility of getting this material? So these slides will be available to you after the presentation online on our website. So you'll be able to take a look at them again once this is over. Another question here was the sensor placed on the myocardial tissue on isolated Langendorf prep, or was it inserted? And so actually the slide that I showed you, the sensor was inserted into the inner chamber, so into the inside of the ventricle, not necessarily in the myocardial tissue, but just in the ventricle itself. And so it was monitoring really the pO2 in the reconstituted blood that they were using to perfuse the heart into the ventricle, and not necessarily monitoring the pO2 of the tissue. But what's great about that is it's completely non-invasive, quote unquote, in that you're not actually puncturing the tissue. So you can get an idea of the sort of oxygen consumption of the ventricle wall and that myocardial tissue without having to put the sensor into it. So a really cool application. I hope that answered your question. Another question here is, can you suggest any references for using the OxyLite in mouse muscle? So yes, we do have a large list of citations that look at ischemia reperfusion type work in small animal muscle. So I showed a rat one, but we do have a ton of citations, so you can peruse our citation list for references there. And we can also offer advice in terms of sites and kind of how to use the OxyLite in muscle. Another question here is, what is the maximum depth that can be measured? So for example, in brain studies, you can get measurements in deep structures like the thalamus. So to answer your question, you can measure at any depth that you want. You just have to be willing to put the sensor there. So if it is an invasive sensor, especially for the pO2. And so you do have to get the sensor to the deep structure. So you mentioned the thalamus. If you can get a sensor into the thalamus in whatever animal model you're using, then yes, you can measure you can measure flow and oxygen there. And you would probably use one of our bare fiber sensors, which is a really thin set of glass fibers that range anywhere from 300 micron or 250 microns up to 450 microns. So depending on your animal model, that might be minimally invasive for what you're working with. Someone's asked if there is limitations to the OxyLite OxyFlo in cardiac applications. So as I mentioned before, the OxyFlo doesn't work that well in cardiac applications because of the movement artifact and the animal's heart is beating. But the OxyLite does not have any limitations except getting the sensor where you want it. So again, with a beating heart, puncturing the myocardium and putting the sensor into the tissue can be really tricky in really small animals like mice. We do have customers who use the OxyLite with the bare fiber sensors, those really thin, small fiber sensors that I mentioned, in rat myocardium and mouse myocardium, but you definitely require like a surgical finesse for the insertion techniques. And we can offer advice on that as well, if that's something that you're interested in. Another question here is, you talked about tissue vitality. Can a tissue be vital if O2 is high but perfusion is low? So I guess theoretically, yes. It really depends on what kind of tissue you're talking about. I will share an example of some of my experience if I can, and hopefully it will kind of answer your question. So the particular experimental design for this group was resuscitation model, and we were monitoring perfusion in the brain after prolonged cardiac arrest, and then with CPR to resuscitate the animal. And something that we noticed was that through the prolonged cardiac arrest and essentially cessation of blood flow, of course the flow dropped and obviously the tissue dropped following that, but with resuscitation, flow came back, oxygen came back, and it actually overshot. So the oxygen was being delivered to the tissue, but the brain in the region that we were looking at was very likely damaged and injured to the point that it was no longer consuming oxygen. So the source and the sink, the supply and the demand in that tissue were not balanced anymore. And we actually saw an increased oxygen concentration in the tissue, even though it likely appeared to be damaged. And so it depends, again, on your application, but you can have high O2 and a tissue not really be vital. So I hope that answered your question. So I guess the answer there is potentially, depending on the tissue of interest. You can also have high oxygen in areas near the lungs and near large arteries that aren't necessarily like functional tissues that are vital, but they still have high oxygen concentrations because of their location. And that the oxygen is being delivered readily, but not necessarily being consumed at its intended rate. So I hope that answered your question. Okay, so another question here is, we generally think of blood oxygenation in terms of oxygenated hemoglobin, but the OxyLite is detecting free oxygen. What is the correlation between those? I didn't think there was much free oxygen in the body / blood. So this is a like textbook question that we get a lot. The OxyLite is detecting dissolved oxygen, so free oxygen. And so if you were to place an OxyLite sensor in the blood, for example, that is also being monitored with pulse oximetry, for example, you can get 100% saturation of hemoglobin. And with that also comes free oxygen in the blood. So if oxygen is dissociating from hemoglobin to be delivered, eventually that oxygen has to come off of the hemoglobin and perfuse into the tissue. And so that's sort of how oxygen diffusion works. It's not really being delivered, hand delivered, if you will, from the hemoglobin to the cell of interest. Really, it's dissociating from the hemoglobin, diffusing into the tissue, and then the cells from there are picking it up. So there is actually quite a bit of free oxygen in the body. And by quite a bit, I mean, that's a relative term too. You don't necessarily have, 150 millimeters of mercury in any given tissue. For reference, anyone who is kind of familiar with how pO2 works, partial pressure of oxygen in the air can be measured with the OxyLite, which is really cool, except that that's going to be way above any sort of in vivo tissue oxygen tensions. And so in vivo, really when the animals are breathing 21% oxygen, some of the highest pO2s that we see are about 90 millimeters of mercury. And you don't see any sort of really hyperoxic tissues unless they're breathing 100% O2, like pure oxygen, basically. And so there is more free oxygen in the body than you would think, but it's not to the extent that we have it in the air. So I hope that kind of answers your question. But one really cool option with the OxyLite is sort of whenever you get the sensor where it needs to be, it will measure. It's not site specific or application specific. It will just measure the pO2. So you can measure it in the air, you can measure it in a glass of water, you can measure it in a cell culture, but you can also measure it in the tissue. And so those reference values from any of the examples that I mentioned will kind of show you which tissues are highly perfused, which ones have a lot of oxygen, and which ones consume a lot of oxygen as well. When you do an ischemia reperfusion type study and you clamp the artery or you cut off the supply, how quickly does the reserve diminish, and basically how high is that demand? And in most vital organs, it's a pretty high demand, especially the brain. And muscle that's functioning, like cardiac muscle, which is always functioning if the animal's alive. So I hope that answered your question. Sorry, I rambled on. But if you do have any more questions about that, you can also send an email and we'll try and get to them. Another question here is, does the pO2 sensor or can it be used to track chronic pO2 changes? So the answer to that is yes. In some applications, you can use our chronic implantable sensors. So they're mostly used in the brain. And if you watch the video on our website, it explains them a little bit better. But essentially, it's a sensor that you implant and you just have to leave a small little end of the sensor outside of the body so that you can connect it to our adapter cable. And so we do have people that use these in small animal brain studies where they just basically can monitor pO2 changes in the brain while the animals are awake and moving around. And you just sort of kind of plug them into the adapter cable and they walk around with this little tether on their head to monitor pO2 changes in vivo, conscious, awake animals. But these are really more suited for neuro applications because you need a place to fix that sensor. And mice and rats, if you're familiar with them, they love to chew on each other's appendages and things like that. So definitely singly housed mice for those chronic implantables so they don't chew on each other's sensors. So yes, the short answer to that question is yes, you can track chronic pO2 changes. We do have large animal users that implant full sensors, so not the chronic implantables, but the standard acute sensors. They implant them fully, but in large animals like sheep, to be able to monitor the pO2 changes in the kidney. So they do a hemorrhagic shock model. So they monitor over the course of a couple weeks and the progression of the septic shock and things like that. So yeah, really cool application. So yes, you can track pO2 changes chronically, depending on your application. So I will say that it does depend on your application. Someone else has asked, can the OxyLite be used for blood pO2? And can it be used in the lumen of the GI tract? So the answer to those questions are both yes. When we do insert sensors into arteries and veins, we do like to heparinize the sensors so that they don't clot. But they can be used in the blood to monitor free oxygen in the blood, which is really cool, and a great way to kind of compare the pulse ox technique as well. And then it can also be used in the lumen of the GI tract. So like I mentioned, anywhere that you can get the sensor, it will measure. So you can insert this invasively into the GI tract. It kind of depends on your animal model. And if you try to do it non-invasively, like with an endoscope, that's going to be pretty tricky because the sensors are kind of small and fragile, especially the bare fiber ones. But there probably are some of our sensors that can be used endoscopically and put into the GI tract. So definitely something that we can discuss with you further if you have an interest in the OxyLite for GI work.
Sarah, I
Cool.
think we have time for one more question,
Okay.
if
Yeah,
you
I was
want.
just going to ask. Okay, so the last question is, are the sensors / probes disposable, or is there a cleaning and sanitation process? So the sensors are obviously disposable. You can get rid of them, but they are reusable. So the OxyFlo sensors can be reused forever if you take really good care of them. The OxyLite sensors are consumable but reusable. So earlier in the presentation, I showed you how they work and kind of what they look like up close. That phosphorescent dye can be photo bleached and will be photo bleached with continuous use. And we do have specification information on exactly how long the sensors can be used for before the dye is photo bleached. But there are cleaning procedures that can be used to be able to use them regularly and in different animals. They don't come sterile, because you did ask about a sanitation process. So just to kind of cover another question here about the sterility of the sensors. So they're manufactured in a clean room, but they're not sterile. However, you can sterilize them using non-heat procedures like STERRAD or ethylene oxide, and we can recommend protocols for sensor sterility if that's something that you require. It does affect the lifetime of the probe, however, can be done, and we do have customers who do sterilize their probes. So I hope that answered your question. All right, so yeah, I think we're out of time here.
Yeah, unfortunately, we had a lot of questions that we couldn't get to, but we will get to those at a later time. And thank you again, Sarah, for wonderful presentation and explanation of this system and these applications. And we'd like to thank you guys, of course, for attending. So we've reached the 1 hour mark, and we really want to be respectful of everyone's time, so we are going to wrap things up. As mentioned, we will answer all these questions privately to those who have asked, and if anyone still would like to ask more questions, they could always contact us. I'd like to finally thank Sarah again for her interesting, incredibly informative presentation, and I'm hoping that all of you walked away satisfied that we met your learning objectives. Please feel free to reach out to us directly if you have any questions about the methodology used to acquire the in vivo data that was presented by Sarah. And thanks again for taking this time out of your day to attend our session. Please look forward to any webinars that we have, and if you'd like to know which are those, please visit our website at scintica.com/webinars. Have a great day.